Welcome to MLSQR. We present part 2 of urinalysis reagent strip analysis, covering urobilinogen, bilirubin, nitrite, and leukocyte esterase testing. We'll cover background clinical lab science, urine chemistry, test methodology, and interfering substances. Afterwards, we'll go over many ACP type question and answers that will help your knowledge and understanding of these interesting topics. First, let's quickly review our previous UA videos. If you need more detail, please see these other videos. Now for the rest of our urine reagent strip reactions and interfering substances. How does urine urobilinogen and bilirubin form? Bilirubin is a highly pigmented yellowish compound derived from degradation products of hemoglobin in the liver and spleen, recycling iron, protein, and protoporphyrin, with some protoporphyrin converted to bilirubin, which is conjugated with glucuronic acid by glucuronal transferase enzymes in the liver to form bilirubin diglucuronide. Bilirubin is reduced by intestinal bacteria to urobilinogen, which is oxidized and excreted in feces as stercobilinogen and fecal urobilin. In the urine, we see hemoglobin degradation products as urobilinogen. However, when the liver, bile ducts, or gallbladder is obstructed and has a disease, water soluble conjugated bilirubin rises in the circulation, which produces jaundice, seen in blood as icteric serum or plasma measured high, known as hyperbilirubinemia. Bilirubin seen in the urine is bilirubinuria. The urine reagent strip urine bilirubin methodology is a diazo reaction with 2,4-dichloroaniline diazonium salt to produce an azo dye. Ictotest by Siemens is a simple and quick confirmatory tablet test for urinary bilirubin using a unique solid diazonium salt. This diazo dye has interference with prolonged light exposure and high doses of vitamin C, and they will cause a false negative. Urine urobilinogen method uses a modification of the Ehrlich reaction. Urobilinogen reacts with dimethylamino benzaldehyde, and we see a color range from light to dark pink. Prolonged light exposure will lead to photooxidation of urobilin, which may lead to a falsely low reading of bilirubin and urobilinogen on the reagent strip. For your information only, nitrifying bacteria converts ammonium to nitrites and nitrates. Here we see a simplified sketch of how nitrifying bacteria plays a role in the nitrogen cycle. What is the clinical significance of a positive urine nitrite test? The clinical significance of urine nitrite testing is that it is a method to detect nitrifying bacteria in the urine, which may be the cause of a urinary tract infection. Urine nitrate testing does not prove or confirm a urinary tract infection, but it simply offers a clue for further investigation. Nitrite positive urine is a sign of nitrifying bacteria that is linked to a urinary tract infection. Nitrates in the urine is caused by bacteria turning nitrates to nitrites, resulting in nitrituria. Note that not all UTIs are caused by nitrifying bacteria. In fact, nitrifying bacteria are not typically found in the urinary tract infections. Common urinary tract infection pathogens are listed here. Which of these are nitrifying bacteria? In one study, only one-fourth of the UTIs had a positive nitrite test. So we see the nitrite test has limited usefulness in the diagnosis of a urinary tract infection. The key ingredient for urine nitrite testing is para-arsenic acid. Nitrate produced by urine bacteria reacts with para-arsenic acid to form a pink azo dye. Exposure to humid air will create false positives on the urine reagent strip for urine nitrite, and it is one of the first to change color from cream to pink. As you might guess, leukocyte esterase is an enzyme found in white blood cells. Esterase is found in neutrophils, eosinophils, basophils, and monocytes, but not in lymphocytes. Together with the nitrite test, leukocyte esterase is a good screen for urinary tract infections and inflammation. Leukocyte esterase catalyzes the conversion of an acid ester indoxyl carbonic acid, and adding diazonium salt, it produces a purple azo dye. False positives for leukocyte esterase include oxidizing agents, formalin, nitrofurantoin, which is highly pigmented, and the false negatives, a high concentration of protein, glucose, oxalic acid, ascorbic acid, and certain drugs. A positive leukocyte esterase and negative nitrite test may indicate one of two things. One, non-nitrifying bacteria, such as those we see in an earlier slide. Or two, the urine in the bladder did not incubate long enough for the nitrifying bacteria to react. The following questions and answers are similar to the ones in our ACP Board of Certification Study Guide. Thank you for your time, attention, and support, and we hope to see you in the next video.